Hello, welcome to the practical number 5, which is about microscope and morphology and structure of the bacteria, taught by Ajahn Lakhana Kanhayiwa Wingfield. Before we move on to the practical, I would like to tell you the objective of this practical. First, you should be able to describe the components of the microscope and use the microscope correctly. Second, you should be able to prepare bacteria for microscopic viewing with emphasis on different types of dyeing, especially gram staining. Third, you should be able to use a microscope to study morphology, gram staining, and other structures of the bacteria. And the fourth, you should be able to describe the colony of the bacteria on the agar plate. Let's move on to the first practical. Microscope. In microbiology laboratory, microscopy is needed to help study the shape and structure of bacteria, fungi, and other microorganisms. A microscope which is commonly used in microbiology labs is bright field microscope. It is a compound microscope which consists of 2 or more lenses. Components of the bright field microscope are shown in the figure. Let's have a look at the first part, eyepiece or ocular. Eyepiece or ocular lens is a lens near the eyes of the user. This lens serves to magnify the objective image. The eyepiece usually has a magnification of 10 times. Body tube or observation tube. Objective lens. Objective lens is a lens available in 3 sizes, which are low power magnification or 10x, high power objective magnification or 40x, and oil immersion objective or 100 magnification. The next part is the revolving nose piece, which you will touch this area to adjust your magnification of the lens. Arm or limb. Coarse adjustment knob is a knob that used to move the camera or tube or state up and down to adjust focus. Fine adjustment knob. Its function is the same as the coarse adjustment, but make the tube or stage move very slowly, making it easier to adjust the focus point. Stage. Stage is a rectangular platform for placing slides or items that need to be viewed with microscope. Mechanical stage. Iris diaphragm. It is used to control the light that passes through the microscope as much or as little as needed. Condenser. Condenser is used to control the light that passes through the microscope. Light source. Light source is the source that will give you the light that it will pass through your specimen. And base. Practical 1. I would like you to be able to use, set up, and maintain the microscope properly. Before performing this experiment, I would like to encourage you to watch the video of how to use, set up, and clean up the microscope, and study the video carefully, and then come back to the lab to get your practical materials, which are microscope, lens wipes, immersion oil, and sample slides. I will be giving you the permanent slides of the bacteria, which are Bacillus subtilis and Escherichia coli, and also the permanent slide of the yeast. Please be careful that immersion oil should be applied when using 100 magnification only. And after you finish observing all the slides, I would like you to record and draw bacteria and yeast that you observe under the microscope, and also describe their shape, size, cell arrangement, and gram staining. Let's move on to the next practical. We are going to study and also view the morphology of the bacteria. For identification of bacteria, you will have to use macroscopic morphology and also microscopic morphology for identification of the bacteria. For this slide, I would like to introduce you about how the bacteria grow on the agar plate. And these are the sample of the bacteria that are growing on the nutrient agar plate. You can see that different type of bacteria, they have different type of color, bacteria, colony size, shape, or even the margin and also the surface of the colony as well. For this practical, I would like to observe the colony of each bacteria carefully. You can use this figure to record the result on your table. Let's have a look at the first bacteria, which is Staphylococcus aureus. But you can see that when we have a closer look at the colony, the shape of the colony is round and the color is like creamy to white in color, or it could be like yellow to cream in color. Here is the colony of the Staphylococcus epidermidis, Enterococcus faecalis, Bredans streptococci, Moraxella, Bacillus subtilis, Escherichia coli, and Proteus vulgaris. For these experiments, I would like you to look at the laboratory report form. You will need to observe the bacteria colony on the plate and then record the size, the shape, and color and other characteristic of each bacterial colony on your table. Apart from observing the bacteria using the macroscopic observation by using our naked eye to observe its colony appearance, microscopic observation is very important for identification of the bacteria as well. Microscopic study are the first step in identifying bacteria, which can be divided into two groups according to staining effect, which can be divided into gram-positive bacteria and gram-negative bacteria. Morphological features include cell shape, which are cocci, bacilli, and spiral, and cell arrangement, including unique structures such as endospore, flagella, capsule, and intracellular granule. As you can see from this figure, that when we view the bacteria under the microscope, you can observe different types of bacterial shape. For example, the round shape like here, or circular shape, we call cocci. And for the rod shape or long shape or elongated shape, we call bacilli. And some of them can have the shape like screw, which is called spiral. And some other, they have a very unique shape and structure. For the cell arrangement, you can see that some bacteria, they are arranged in pair, which is called diplo. Strepto, they will be arranged in chains. And Staphylo, which is called the bacteria that have the cell arrangement as a cluster. For viewing the bacteria under the microscope, we can use different type of staining method, which will facilitate you to view the structure of the bacteria according to your aim. Like if you want to see the spatial structure, you will need to use this type of staining or another type of staining. So let's have a look at the first group of the staining, which is simple staining. Simple staining is a bacterial staining using only one dye. There are two aspects of the bacterial staining, depending on the color charge, with positive staining and negative staining. You can see the positive staining. The dye will stain bacteria into blue color. And for negative staining, the dye will not stain the bacteria in color, but it will dye the background instead. The next type of, The next type of the staining is the differential staining, which can be divided into different methods. I'm going to introduce you about the gram staining. Gram staining is a dyeing technique for basic microbiological analysis. It is used to classify differences of cell wall structure of bacteria that causes different color staining. From the staining results, bacteria that can be divided into two groups, as are shown in the photo. In here is a mixed culture, so you can see two colors of the bacteria. The violet stain is the gram-positive bacteria, and the pink stain is the gram-negative bacteria. So from here, you can also observe the shape of the bacteria. For the gram-positive bacteria, they are cocci, and for the gram-negative bacteria, they are in rod shape. And here is the example of the gram-positive bacteria of the Viridans streptococci. They've got cell arrangement in chains, and their shape is cocci. And for the E. coli, they are stained in red color, which means they are gram-negative bacteria, which has rod shape. For gram staining, as I told you, that is the basic dye and is very important in the study of bacterial morphology. This staining can divide bacteria into two groups, as I told you before. For the gram-positive staining, for the gram-positive bacteria, which attach to the crystal violet color, and for the gram-negative bacteria, which is attached to the color of the safranin. Acid-fast staining. Acid-fast staining is a dyeing technique used to detect bacteria that causes tuberculosis. Microbacterium tuberculosis is stained with the red color of the carbofuchsin, while other bacteria and cells are stained with methylene blue, as shown in the picture. Let's have a look at the first figure. The acid-fast staining organism will show in the bright red to purple in color. And for non-acid-fast bacteria, you will see them in the blue color. For the TB or Mycobacterium tuberculosis, you can see they will be stained in red color here. And you can also observe their structure. And for non-microbacterial bacteria, for example, Nocardia, you can see them in the blue color. Apart from those staining methods, there are some other methods that we normally use to study the spatial structure of the bacteria. The first one is the flagellus staining. Some bacteria have flagella, which requires a special technique called flagella staining method to observe the structure. Stained flagella of the bacteria are shown in the picture. Spore staining. Bacteria in the genus Bacillus and genus Clostridium can transform the cell structure for survival to the endospore-forming bacteria when the environment is not suitable. Spore staining method is used to observe endospore in the cell, as shown in the figure. That you can see that the color of the endspore is stained in green and the cell is stained in pink. Capsule staining. Some bacteria have capsule around the cell, which requires a special technique called capsule staining method to observe the capsule. Many methods of negative stains are commonly used to clearly detect cells surrounding the capsule as well. In this practical, we are going to focus on the gram staining method. Here are some examples of the gram staining. I would like to encourage you to watch the video of the gram staining prior to performing the experiment. And here is just a brief introduction of how to perform the gram staining. And please also be careful that aseptic technique is required all the time. Briefly, for gram staining of the bacteria that grow in the liquid media, you can take 2 loops of the sample and then smear onto the slide and try to confine the sides of the smear area into 1 centimeter, and then perform the air dry. And after the sample are dry, you have to fix it with flame using the forceps and do the fixation for 3 to 5 times. And then move on to the gram staining area, which I provide you at the sink close to the window. And after you finish the staining, you need to absorb all the leftover water, and then your slide will be ready to be observed under the microscope. And here are the instructions of how to do or perform the gram staining. First, after you finish the fixed dyeing of your slide, you will have to apply crystal violet, which is a purple dye, for 30 seconds, and then watch. Then apply the iodine, which is a mordant, for 30 seconds, and then wash. Then apply alcohol, which is a decolorizer, just in a short time, like 3 to 10 seconds, and then wash. And then apply safranine, which is counter stain, for 15 seconds, and then wash. As you can see from this diagram, in each step, it will show the different staining of the bacteria according to the cell component. And again, I would like to encourage you to watch the video before performing this practical. Okay. For this practical, I will divide it into 2 sections. The first section is I would like you to practice performing the simple staining. Simple staining, we will be giving you the yeast sample, which is Saccharomyces cerevisiae. Then you will have to prepare the slide of the Saccharomyces cerevisiae, and then you do the smear and then perform the fixation. But actually, you should not perform heat fix for too long for this type of organism. And then you add 1 drop of the methylene blue and then wait for 1 minute and then wash. And then you can observe the yeast structure under the microscope. I would like to suggest you to use the 40 magnification to observe the yeast structure, because the yeast structure is much bigger than the bacteria. So it is not necessary to perform the immersion objective. And for the second practical, I would like you to do the gram staining of the bacteria. I will be giving you gram negative and gram positive bacteria. For this experiment, actually, I will not tell you which one is the gram positive and gram negative bacteria, but I would like you to observe them by yourself. For the Staphylococcus aureus, I will be providing you in a plate. And for the Bacillus subtilis, it will be grown on a slant agar. For Streptococcus pyogenes, it will be grown in the broth culture. And for the Escherichia coli, it will be grown on a plate as well. Okay, after you take all those bacterial sample, you perform the gram staining, smear fixation. For one student, I would like you to do 2 samples on one slide, and also don't forget to label the name of the bacteria on it. And here are the image that I am, that I wish that you could be able to see under your slide sample. Here is the structure of the, Streptococcus pyogenes. Cell arrangement is in chain, and it is gram positive bacteria, and the shape is cocci. For E. coli, it is gram negative bacteria. The shape is rod and no cell arrangement. For Bacillus subtilis, they are gram positive bacteria in rod shape. And I would like you to look carefully. Sometimes you can also observe the endospore structure in here, as I show you here in the arrow. And for the Staphylococcus aureus, they are gram positive bacteria as well, which is cocci, and the cell arrangement is in cluster. For the bacteria, you can use the immersion oil to observe their structure clearly. So these are all the experiments for our practical number 5. I hope that you'll be able to differentiate between gram positive and gram negative bacteria, and you should be able to perform gram staining professionally, probably, and you should be able to observe the color, shape, and cell arrangement of the bacteria given. After you finish the experiment, you will have to look on your lab report. For this one, I would like you to observe the bacterial structure after staining and then record the color of the bacteria, and also identify if they are gram positive or gram negative bacteria. And also observe the shape of the bacteria, round, rod, or spiral. You should also observe the cell arrangement in chain, cluster, or in pair, and other structure if you can see, for example, endospores. And also don't forget to record colony characteristic of each bacteria grown in NA as demonstrated.